I want you to picture something for me. Imagine you are standing in front of a library. But this isn't just any library. This is the most secure, fireproof, historically significant archive in the existence of the universe. It is built to last forever. And inside the central vault of this library, you have the original blueprints for everything. Every machine, every tool, every structural beam needed to build a massive, sprawling, living civilization.
That is the DNA, sitting safely inside the nucleus, the ultimate instruction manual.
Exactly. It's valuable. It's essential. You cannot build a civilization without it. But here's the problem I've been thinking about while reading through the chapters for today's deep dive. A blueprint cannot build a wall. A blueprint cannot fight off an invader. A blueprint cannot digest your lunch. If you just have a stack of papers, nothing happens.
Precisely. Information by itself is inert. It is just potential. It sits there. For life to actually happen, for breathing, moving, thinking, fighting infection, that information has to be converted into action. Yep. And in the biochemical world, action is almost synonymous with protein.
And we've spent a lot of time in previous deep dives talking about the library, the DNA, and the photocopies of the blueprints, the RNA. That's the central dogma we all learn in school. DNA makes RNA, RNA makes protein. But today we are stopping the photocopier. We are walking out of the library, and we are going to the construction site. We are finally doing the deep dive into protein metabolism.
The point where the plan meets the pavement.
Right. And I have to say, looking at these sources, we are pulling from Leninger's Principles of Biochemistry for the systems logic and Harper's Illustrated Biochemistry for the clinical side. I actually thought this was just going to be about how proteins are made. You know, bead A connects to bead B, make a necklace, done.
And I assume you found out pretty quickly that it's a little more complicated than bead A meets bead B.
A little. It's a logistical nightmare. It's not just manufacturing, it's shipping, it's quality control, it's security clearance, and it's waste management. It feels less like a biological process and more like we're describing a massive bureaucratic city.
That is actually a perfect analogy, and I'm glad you landed there. The theme we really want to explore today is governance in motion. Because protein metabolism isn't just synthesis, it is a life cycle. It involves incredibly strict quality control, precise targeting like a global postal system, and planned obsolescence. The cell is obsessed with fidelity. It wants to know that the right worker is in the right place at the right time doing the right job, and when that job is done,
it gets terminated.
Precisely. It is a ruthless system. This isn't just about building, it's about managing a dynamic workforce.
So to help us navigate this, we're going to adopt a bit of a persona today. The outline calls it Medlock Holmes. We are going to be investigators following the trail of genetic information to its physical outcome. We're asking the big questions. How do we translate a 4-letter language into a 20-letter language? Why does the cell spend so much expensive energy doing it? And this is the part that really grabbed me. What happens when the bureaucracy fails?
And that's where Harper's really shines, bringing in the clinical side. We'll see that diseases like cystic fibrosis or Alzheimer's are, at their core, failures of this governance system. But to understand the failure, we first have to understand the logic. We have to start with the language itself.
The logic of the code. Let's start there. This is part 1. The rules of translation. Before we get to the machines, we have to solve a math problem. We know DNA and RNA have a 4-letter alphabet, A, C, U, G. But proteins are built from a 20-letter alphabet, the 20 standard amino acids. So you can't just have one base equal one amino acid. It's a simple numbers game.
Right. If you had a 1-to-1 ratio, you'd only be able to come for 4 amino acids. That's not enough to build life as we know it.
Okay, so what about pairs? A 2-letter word, say AC or GU.
Well, with 4 letters, the number of possible 2-letter combinations is 4 squared. That's 16. Still not enough. You'd be short 4 amino acids, and you wouldn't have any punctuation, like a stop signal.
So nature went with 3, the triplet codon.
Yes, exactly. 4 cubed gives you 64 possible combinations. That's plenty. It covers the 20 amino acids and leaves you with some extras for punctuation. 3 stop codons, UAA, UAG, and UGA, and 1 start codon, AUG.
But this immediately creates a new situation. We have 64 possible words, but only 20 things to name. This leads to something Leninger calls degeneracy, which sounds like an insult, like the genetic code has a gambling problem, but in biochemistry, it's actually a safety feature, isn't it?
It is a massive safety feature. Degeneracy just means that the code is redundant. It's not a moral failing, it's a design choice. Since we have 61 codons that code for amino acids, but only 20 amino acids, most amino acids have multiple phone numbers that reach them.
Multiple codons for the same output.
Right. For example, leucine and serine are very common amino acids. They each have 6 different codons that all code for them. Arginine also has 6.
6. So CUU, CUC, CUA, CUG. They all mean leucine.
Correct. And think about why that's brilliant from an engineering perspective. DNA is constantly being damaged. UV light, chemical mutagens, simple copying errors. A single letter change of point mutation is a common event.
And if that change happens in the third position of the codon,
There's a very high chance it won't matter. If you look at the genetic code table, you see the redundancy is often clustered in that third base. So a mutation changing CUU to CUC still codes for leucine. The mutation becomes silent. The protein is made correctly. It's a buffer against error.
it's like a buffer. If I misspell a word slightly in an email, you can usually still figure out what I mean. But Leninger makes a very specific distinction here that I want to unpack. The code is degenerate, but it is unambiguous.
That is a crucial distinction. It's maybe the most important rule.
Yeah.
Unambiguous means that a specific codon, like CUU, will only ever code for leucine. It never gets confused and accidentally codes for serine or alanine or anything else.
So one amino acid can have many codes.
But one code can have only one amino acid meaning. It's like a language where a word might have synonyms, but no word has a double meaning. It ensures precision. There's no room for interpretation at the ribosome.
And it's continuous, right? The outline says punctuation free. There are no commas, no spaces.
None at all. Once the ribosome finds the start line, the AUG codon, which sets the reading frame, it reads every 3 letters, nonstop, like a train on a track, until it hits one of those 3 stop codons.
This is why frameshift mutations are so devastating. If you insert or delete just 1 or 2 letters, everything downstream shifts over. It completely garbles the message.
It turns the rest of the message into total gibberish. It's like taking the sentence, the fat cat sat, and deleting the H. You get tef et hera atsat. It means nothing. And usually you'll hit a premature stop codon and the protein just truncates. It's a catastrophic failure.
No, generally we're told this code is universal. If I look at a bacteria, a banana, or a human, AUG means methionine.
Generally, yes, and that's profound. It's one of the strongest pieces of evidence for a single common ancestor for all life on Earth. The operating system is conserved across kingdoms, but there are exceptions. And interestingly, they are mostly inside you right now, but not in your nucleus. They're in your mitochondria.
The mitochondria, the powerhouse, but apparently also the rebel of the cell.
A bit of a rebel. Harpers points out that human mitochondria read the code slightly differently. It's a fascinating evolutionary quirk. For example, in the standard universal code, the codon UGA is a stop codon. It means stop working. In our mitochondria, it codes for the amino acid tryptophan.
That seems like a dangerous mix-up. If you put mitochondrial genes into the main cell, the machinery will just stop halfway through.
I would. And another example is AUA. In the cytoplasm, it means isoleucine. In the mitochondria, it means methionine.
So why the difference? Why this separate dialect?
It comes down to efficiency and evolution. Mitochondria are thought to have originated from free-living bacteria that were engulfed by an early eukaryotic cell. They have their own tiny circular genome and their own separate translation machinery. Over billions of years, they've streamlined the process. They have fewer transfer RNAs, tRNAs available, only about 22 compared to the 31 or so in the cytoplasm. So they've compressed the code, reassigned some codons to work with fewer resources. It's a high-efficiency, stripped-down operating system.
Speaking of efficiency, that brings us to the wobble hypothesis. I love the name. It sounds like a dance move from the 60s, but it's actually about how the cell saves money on tRNAs.
It is. It's a concept from Francis Crick, actually. We have 61 codons that code for amino acids. Theoretically, you'd expect to need 61 distinct tRNAs, the little adapter molecules that carry the amino acids to match them perfectly. But most cells don't have that many. They get by with far fewer.
Because the third base is allowed to wobble, it doesn't have to be a perfect match.
Exactly. The first 2 bases of the codon on the mRNA form strong, standard Watson-Crick base pairs with the tRNA's anticodon. They are the strict identifiers. They have to match perfectly. But the pairing between the third base of the codon, the one at the 3-bit end, and the first base of the anticodon at the 5-bit end, has a bit more geometric freedom. It can wobble.
It's a looser fit.
It's a looser fit, allowing for non-standard base pairings. And Leninger mentions a special nucleotide here that makes this even more efficient. It's called inosine. Yes, inosine. It's a modified purine nucleotide found in some tRNAs, specifically in that first wobble position of the anticodon. It's incredibly promiscuous, chemically speaking. Inosine can form hydrogen bonds with uracil, cytosine, or adenine. It can't pair with guanine, but it covers the other 3.
So if you have a single tRNA with an inosine in the wobble position, that one tRNA can recognize and bind to 3 different codons.
Precisely. For example, the codons for alanine are GCU, GCC, and GCA. Instead of making 3 different tRNAs, the cell can just make one that has an inosine in the wobble position of its anticodon. That one tRNA can read all 3 codons.
So the cell doesn't need to build a separate truck for every single delivery address. It has master keys that can handle a whole neighborhood. It's an economic strategy.
It's pure biochemical economics.
Uh-uh.
Minimize the number of parts you have to build while maintaining the integrity of the information.
Okay, so that's the code. We have the rules of the language. Now we need to visit the factory floor. We need to talk about the machinery of fidelity, because this is where the Medlock Holmes investigation really starts to get interesting. We have the tRNA, which acts as the adapter. It reads the code on one end and holds the amino acid on the other. But here's the part that blew my mind. The ribosome, the massive machine that builds the protein, is blind.
It is utterly blind to the cargo. The ribosome has no way of checking if the tRNA is holding the correct amino acid. It only checks the barcode, the anticodon on the tRNA. If the anticodon matches the codon on the mRNA, the ribosome accepts it. If a tRNA that's supposed to carry serine accidentally gets loaded with glycine, the ribosome will just put the glycine in. It trusts the tRNA completely.
Which means the actual error checking, the actual translation logic, isn't happening in the ribosome. It's happening before that. It's happening with the enzymes that load the trucks, the aminoacyl tRNA synthetases.
These are the unsung heroes of biology. I would argue they're the true translators. They're the only molecules in the cell that know the genetic code. They have to recognize both a specific amino acid and all the tRNAs that correspond to it. They physically bridge the gap between the world of nucleic acids and the world of amino acids. Some people call this the second genetic code.
The second genetic code. The rules of how synthetases recognize their specific tRNAs.
Right. And they have to be incredibly accurate. The whole system's fidelity rests on their shoulders. And Leninger describes this beautiful double sieve mechanism that I found fascinating. He uses the example of isoleucine and valine. Can you walk us through that? Because chemically, those two are almost identical.
They are. Valine and isoleucine are both branched-chain hydrophobic amino acids. They differ by just a single methylene group. That's 1 carbon and 2 hydrogens. It's a tiny, tiny difference in size and shape. Valine is just slightly smaller than isoleucine.
So how does the enzyme, the isoleucyl tRNA synthetase, tell them apart? It needs to attach isoleucine but reject valine.
It uses 2 filters, or sieves, in 2 different parts of the enzyme. The first sieve is the main active site, the activation site. This is where the enzyme tries to grab isoleucine and attach it to an ATP molecule. The pocket is sized perfectly for isoleucine. Anything larger simply won't fit. So that filters out all the big amino acids like phenylalanine or tyrosine.
Okay, so the big ones are out. But valine is smaller. It can sneak into the isoleucine pocket. Exactly. Valine is an imperfect fit, but it's close enough. It slips through the first sieve. It gets activated. If the enzyme stopped there, it would mistakenly attach valine to the isoleucine tRNA about 1 time in 100. That's an error rate of 1%.
Which sounds low, but for a protein with 500 amino acids, a 1% error rate means on average every single protein would have 5 defects. That's not viable.
Right, it would be catastrophic. So cellular machinery would be riddled with errors. So nature evolved a second sieve. After the amino acid is activated, the enzyme tries to move it to the tRNA. But on the way, it passes a second active site. This is the proofreading or hydrolytic site.
And this one is different.
Yes, this second pocket is smaller than the first one. Isoleucine, the correct amino acid, is too big to fit into this second pocket. It gets stuck, so it proceeds on its way, gets attached to the tRNA. It passes the inspection.
But valine, the mistake.
Valine is small enough to fall into the second pocket. And inside that second pocket is a hydrolysis site. It's a molecular shredder. If an amino acid falls in there, the enzyme immediately cleaves it off the AMP, destroying the mistake. So valine gets sieved out and rejected.
That is brilliant engineering. It's like a coin sorter. The correct coin is the only one that fits in the first slot but is too big for the second slot. Anything that fits in both slots is a counterfeit and gets discarded.
Exactly. And this double-check mechanism is so effective, it drops the error rate from 1 in 100 to about 1 in 10,000. That's the level of fidelity life requires.
But fidelity isn't free. This brings us to the thermodynamics, the energy cost. Leninger makes a point to tally up the bill for making a single peptide bond, and it is expensive.
It's incredibly expensive. You have to remember, forming a peptide bond is thermodynamically unfavorable. You're trying to create order, so you have to pay for it. The cell burns through at least 4 high-energy phosphate bonds to add just 1 amino acid to a chain.
Let's break that down. Where do they go?
First, you have to activate the amino acid. That costs 2 ATP equivalents. The synthetase takes an ATP and breaks it down to AMP and pyrophosphate. That's a huge release of energy used to charge the amino acid. So that's 2.
Okay, 2 to load the truck.
Then, to deliver that charged tRNA to the ribosome, an elongation factor protein uses 1 molecule of GTP. So that's 3. And then, after the peptide bond is formed, the ribosome has to move 1 codon down the mRNA. That's called translocation. Another elongation factor uses another GTP for that step.
So that's 4. 2 from ATP, 2 from GTP, 4 high-energy bonds for 1 link in the chain.
Yes, and that's the minimum. That's for a successful addition. If the proofreading kicks in, like we just discussed with valine, you've already spent the 2 ATP equivalents to activate it, and then you just throw that energy away to destroy the mistake. The cost of an error is even higher.
Why? Why is the cell willing to pay that much? This seems incredibly wasteful on the surface.
Because the alternative is death. Thermodynamics tells us that entropy disorder is the natural state of things. To create order, to create a protein that folds exactly the same way every single time and performs a precise function, you have to pay for it with energy. The cell is buying certainty. It is buying fidelity. A single protein folded wrong can aggregate, cause toxicity, and kill the cell. The energy bill is the non-negotiable cost of doing business reliably.
And that's a perfect segue to part 3, the assembly line. Because synthesis is just the beginning. The polypeptide chain comes out of the ribosome, but it's just a wet noodle. It's an unfolded linear chain of amino acids. It needs to fold into a complex 3-dimensional shape. And I used to think folding just happened. You know, physics takes over, the positive and negative parts attract, the greasy parts hide, magnets click together, done.
For some small, simple proteins in a dilute solution in a test tube, that's often true. That was Anfinson's Nobel Prize-winning experiment. But for larger, complex proteins, the cellular environment is nothing like a test tube. It's incredibly crowded. The cytoplasm is packed with other proteins, RNAs, metabolites. It's like trying to fold a complex origami crane in the middle of a mosh pit.
You're going to get bumped. You're going to stick to the wrong people.
Exactly. Especially the hydrophobic or greasy parts of the protein. They desperately want to get away from water, and their first instinct is to stick to the nearest greasy patch they can find, which might be on another unfolded protein. That leads to aggregation, a big, useless, often toxic clump of protein.
So you need help. You need bodyguards. You need chaperones.
Specifically, the heat shock proteins, or the HSP70 family. They're called that because their production ramps up when a cell is stressed, like by heat, which causes proteins to unfold. These are the first responders. As soon as the new protein chain starts poking out of the ribosome, these HSP70 chaperones grab onto the exposed hydrophobic parts.
They cover up the sticky bits.
They cover up the sticky bits to prevent premature folding or aggregation. HSP70 binds to them, and using the energy from ATP hydrolysis, it clamps on, holds them safe, and then later releases them to give them a chance to fold correctly. It's a cycle of bind and release, giving the protein time and space to find its proper shape.
But sometimes HSP70 isn't enough. Sometimes you need to escalate. You need the big guns. You need the chaperonins. And Leninger has these incredible diagrams of GRO-EL and GRO-A's in bacteria. It literally looks like a barrel with a lid.
It is a barrel. It's a large double-chambered protein complex. And it functions as an isolation chamber. We call it an Anfinson cage, in honor of the guy who showed proteins could fold on their own in ideal conditions. This machine creates those ideal conditions inside the cell.
How does it work? It's not just a passive container, is it?
Not at all. It's an active machine. The unfolded protein, often handed off from HSP70, is captured by the hydrophobic lining at the mouth of one of the barrel's chambers. Then, a lid, a separate complex called GRO-ES, caps the top. This process is driven by the binding of 7 ATP molecules.
7 ATPs, just to put the lid on.
All right, and once the lid is on, the inside of the chamber changes. It becomes hydrophilic, which helps kick the protein off the walls and allows it to float freely inside this tiny private room. It's isolated from the crowded cytoplasm. It can now attempt to fold without bumping into anything else.
Solitary confinement for folding.
Basically. And it's timed. The hydrolysis of those 7 ATPs acts as a timer. After about 10 to 15 seconds, the ATP is hydrolyzed to ADP, the lid pops off, and the protein is ejected. If it's folded correctly, great. It goes off to do its job. If not, it might get rebound and shoved back in for another round.
That visual is wild. Just a protein getting stuffed into a barrel, lid goes on, tick, 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 lid pops off. Are you done? No. Get back in. It's an incredibly energy-intensive process.
It shows how critical correct folding is. But the stakes get even higher when we move from the cytoplasm to a different cellular compartment. The endoplasmic reticulum, the ER, Harper's focuses heavily on this because this is the entry point for the secretory pathway. This is where proteins destined for the cell membrane or for export out of the cell like hormones, antibodies, receptors are made and folded.
The ER sounds like a high-stress, high-stakes environment.
It is. It's an oxidizing environment, which is necessary for forming disulfide bonds, but it's also a crowded factory floor, so it has its own dedicated set of chaperones. One of the most important is called bipie-binding immunoglobulin protein. It's the ER's version of HSP70. As proteins are threaded into the ER, BAP grabs them and acts like a ratchet, preventing them from sliding back out. It also helps with their folding.
And then there are the ones that check the sugar tags, calnexin and calreticulin. That seems like a different strategy.
It's a key quality control checkpoint. Many proteins made in the ER are glycoproteins. They have complex sugar chains attached to them. Calnexin and calreticulin are lectins, which means they are proteins that specifically bind to carbohydrates. They hold on to these sugar tags and don't let the protein leave the ER until it has achieved its fully folded state. They are the ER's gatekeepers. If the protein is still messy, they hold it back for another try.
So it's not just preventing aggregation, it's active quality control. And there are enzymes that actively fix mistakes too, right? Like protein disulfide isomerase, or PDI.
PDI, yes. Disulfide bonds are strong covalent bonds that form between cysteine residues. They are like the rivets that lock a protein structure in place. But sometimes, in the rush of folding, the rivets get put in the wrong holes, forming incorrect disulfide bonds. PDI is an enzyme that comes along, breaks the wrong bonds, and allows them to reshuffle until the most thermodynamically stable, which is usually the correct configuration, is found.
It's an active repair mechanic on the assembly line.
Exactly. And there are others too, like peptidylprolyl isomerase, or PPI, which helps accelerate a particularly slow step in protein folding involving the amino acid proline. It's a whole suite of tools to ensure quality.
Okay, this is incredible. Governance in motion. Now, assuming the protein is finally made and folded correctly, it has to go somewhere. It can't just sit in the lobby. This is part 4, targeting and traffic, the logistics and shipping department.
And this is a massive sorting problem. A typical human cell might contain 10,000 different kinds of proteins. Some of them need to be in the nucleus, some in the mitochondria, some embedded in the plasma membrane, some are packaged up and shipped out into the bloodstream. How do they all know where to go?
The answer, which won a Nobel Prize for Günter Blubel, is the signal sequence.
A molecular zip code, a built-in shipping label.
Right. It's a short string of amino acids, usually at the very beginning of the protein, the N-terminus that dictates its destination.
For proteins destined for the ER, and from there to the Golgi lysosomes or the outside of the cell, this signal sequence is a stretch of about 10-15 hydrophobic, greasy amino acids. As soon as this sequence emerges from the ribosome during translation, a molecular security guard called the SRP, the signal recognition particle, spots it.
And SRP is bossy. It doesn't just guide, it takes control. It grabs the signal sequence and actually pauses translation. It clamps down and tells the ribosome, stop right now. You are making a restricted item. You cannot finish this here in the general cytoplasm.
That's a great way to put it. It acts as an escort. It drags the entire complex, the ribosome, the mRNA, and the half-finished protein over to the surface of the ER membrane. There, it docks with an SRP receptor. Only then is the pause released and translation resumes. But now the protein is being fed directly through a channel, a pore called a translocon, into the ER lumen. This is called co-translational translocation.
So it's being pushed into the ER at the same time as it's being knit.
Yes. This is the main pathway for all secretory and membrane proteins. But compare that to proteins going to the mitochondria or the nucleus. That's usually post-translational. The protein is made fully in the cytosol first.
So it's already a complete chain. How does it get through a membrane then?
It has to be kept in an unfolded state. Cytosolic chaperones, like HSP70, bind to it and prevent it from folding up into a compact ball. Then this unfolded chain is delivered to the mitochondrial surface, where receptor proteins recognize its specific mitochondrial targeting sequence. Finally, complexes called Tim and Tom pull it through the membranes.
Tim and Tom. Sounds like a comedy duo.
Translocase of the inner membrane and translocase of the outer membrane. They work together, using the proton motive force, the electrical potential across the inner membrane, plus ATP, to actively pull the protein through.
So once we are inside this ER Golgi highway system, we enter the world of vesicular transport, the intracellular shipping fleet. Harper's details 3 main types of protein coats for these vesicles: co-PII, co-PI, and clathrin.
Think of them as different colored trucks for different routes. Co-PI-coated vesicles are your standard forward shipping. They take cargo from the ER to the Golgi apparatus. It's anterograde transport, moving things out toward the cell surface.
And CoPI, that sounds similar.
CoPI is return shipping. It's retrograde transport. Sometimes, essential ER-resident proteins, like that bi-P chaperone or PDI enzyme we mentioned, accidentally get swept up in a CoPI vesicle and sent to the Golgi. They have a special retrieval tag on their C-terminus, a sequence called KDEL. The Golgi has receptors that see that KDEL tag, grab those proteins, and package them into CoPI vesicles to ship them back to the ER where they belong. It's a retrieval system.
And clathrin, that one I've heard of.
Clathrin is in the final leg of the journey, or for incoming goods. It handles traffic from the far side of the Golgi out to the lysosomes or the plasma membrane, and it's also famous for its role in endocytosis, bringing things into the cell. Clathrin itself forms this beautiful basket-like lattice of proteins called a triskelion, which helps to physically curve the membrane and pinch off the vesicle.
The mechanics of how these vesicles actually deliver their goods is fascinating too. It's not just bumping into a wall. It's a highly specific process of docking and fusion. And this involves two key families of proteins, RABs and snares.
Right. First, you have to get the vesicle to the right neighborhood. That's the job of the RAB proteins. They are small GT passes that act as address checkers. Each type of vesicle has a specific RAB protein on its surface, and the target membrane has a receptor that recognizes that RAB. This ensures the delivery truck gets to the right loading dock.
Okay, so RAB gets it close, but how does it actually fuse and unload? That's the snares, right?
That's the snares. They are the machinery of fusion itself. You have V snares on the vesicle, the V is for vesicle, and T snares on the target membrane, T for target.
And they act like a winch, right? Or a zipper.
A winch is a great visualization. When the vesicle is close, these long helical proteins, one from the vesicle and a couple from the target membrane, start to interact. They coil around each other, zipping up into an incredibly stable 4-helix bundle. As they coil tighter and tighter, they physically pull the two membranes into extremely close proximity, squeezing out all the water molecules between them until the lipid bilayers become unstable and fuse. The vesicle membrane becomes part of the target membrane, and the cargo is dumped across.
It sounds violent and irreversible.
It is forceful, and the fusion itself is irreversible. But the snare complex is so stable, it requires energy to take it apart for the next round. A protein called NSF, using ATP, has to come in and act like a wrench to untangle the snares so they can be reused. This is actually where botulinum toxin Botox works. It's a protease that cleaves snare proteins in neurons, preventing vesicles with neurotransmitters from fusing with the membrane. No fusion, no signal, no muscle contraction.
Wow. So we've synthesized, folded, and shipped, but nothing lasts forever. And this is where the governance metaphor gets really interesting. Part 5, quality control and degradation. I used to think degradation was just taking out the trash. But Leninger and Harper's both frame it differently. They say turnover is regulation.
Absolutely. This is one of the most important concepts. If you have a protein that acts as a signal, say a transcription factor that turns on a set of genes, or a cyclin that tells the cell to divide, you do not want that protein hanging around forever. You need its action to be brief and timely. You need to destroy it immediately after the signal is received. If you don't, you get a persistent, unregulated signal. Uncontrolled growth, for example, that's cancer. So degradation is about stopping a signal just as much as it is about removing damaged goods.
It's information control. And there's a rule for this. The outline mentions the N-end rule.
Yes, it's a fascinating principle. The stability of many proteins in the cytoplasm is determined by the very first amino acid at its N-terminus. It's like an expiration date stamped on the forehead of the protein.
So if a protein starts with methionine or alanine or glycine,
it's considered stabilizing. That protein might last for more than 20 hours in the cell. It has a long half-life.
But if it starts with arginine or leucine or phenylalanine?
Those are destabilizing residues. A protein with one of those at its N-end has a half-life of just a few minutes. The cell's degradation machinery sees that residue and marks the protein for immediate destruction.
And the mark of death, the tag that says destroy me, is a tiny protein called ubiquitin.
Ubiquitin. It's a small 76 amino acid protein that is, as its name suggests, ubiquitous and highly conserved across all eukaryotes. It is the black spot. The cell has a 3-enzyme cascade E1, E2, and E3 that works together to attach ubiquitin to a lysine residue on the target protein.
Okay, E1, E2, E3. What do they each do?
E1 is the ubiquitin activating enzyme. It uses ATP to activate a ubiquitin molecule, forming a high-energy bond. E2 is the ubiquitin conjugating enzyme. It takes the activated ubiquitin from E1. Think of E1 as the bank that provides the activated ubiquitin, and E2 as the armored car that transports it.
So who's the hitman? Who picks the target?
That's E3. The E3 ubiquitin ligase is the brain of the operation. There are hundreds of different E3 ligases in the cell. Each one is specialized to recognize a specific target protein or a specific signal, like the N-end rule residue. The E3 brings the E2 with its ubiquitin cargo and the target protein together and catalyzes the transfer of ubiquitin onto the target. The specificity of the whole system lies with the E3s.
And it's not just one ubiquitin molecule, is it?
No. For a protein to be sent for degradation, it needs to be tagged with a chain of at least 4 ubiquitin molecules linked in a specific way. This is polyubiquitination. It's an unambiguous destroy-me signal.
And once you get tagged with that chain, you are sent to the proteasome.
The 26S codisome, the execution chamber, the protein recycling center.
Leninger describes this structure in detail. It's another one of these barrel-shaped complexes, isn't it?
It is. The core of it is the 20S particle, which is a hollow cylinder lined on the inside with protease active sites. The blades are on the inside, which is critical. It protects the rest of the cell from being randomly chopped up. But the crucial part is the 19S caps that sit on either end of this barrel. These are the regulatory gates.
What do the gates do?
They do 3 things. First, they recognize the polyubiquitin tag on the doomed protein. Second, using a lot of ADP, they unfold the protein. They have to linearize it, stripping it straight. And third, they thread the unfolded chain into the core of the proteasome.
Like feeding a log into a wood chipper.
Exactly. It's an active, energy-dependent process. Inside the core, the protein gets chopped into tiny peptides, about 3 to 25 amino acids long. The ubiquitin molecules are cleaved off and recycled, and the resulting amino acids are also reused by the cell. It's the ultimate recycling center.
But what if the problem protein is in the ER? We talked about those chaperones trying to fix things. What if they fail? You can't just have a proteasome inside the ER, can you?
No, you can't. The proteasome only lives in the cytoplasm and nucleus. So if a protein in the ER is terminally misfolded, and the chaperones like calnexin and BP can't fix it, they give up. The cell then initiates a process called ERAD-ER-associated degradation. The protein is literally pushed back out of the ER through a translocon into the cytosol.
It gets ejected.
It gets ejected.
Yeah.
And as it emerges into the cytosol, an E3 ligase is waiting. It immediately tags the protein with ubiquitin, and it's sent off to the proteasome for destruction. The cell is essentially taking out its trash.
But what if there's too much trash? What if the system is overwhelmed with misfolded proteins?
Then the cell declares a state of emergency. This is the unfolded protein response, or UPR. When misfolded proteins pile up in the ER, they bind to chaperone proteins like BiP. This pulls BiP away from sensor proteins that are embedded in the ER membrane. When BiP lets go, these sensors become active.
And they send out an alarm.
They send alarm signals to the nucleus and the cytoplasm to do 3 things, basically. First, shut down most new protein synthesis. Stop the assembly line to reduce the load. Second, ramp up production of more chaperones and folding enzymes. Get more workers to deal with the backlog. And third, increase the machinery for ERAD. Get more trash cans.
It's a surge strategy to restore balance or proteostasis.
It is. But if the surge fails, if the stress persists and the cell can't clear the backlog, the UPR switches its programming. It shifts from a save the cell response to a kill the cell response. It activates pathways that lead to apoptosis or programmed cell death. The cell commits suicide to protect the organism from being damaged by a rogue malfunctioning factory.
Which brings us directly to part 6, clinical relevance. Because when this governance system fails, in either direction, we get disease. And Harbers highlights cystic fibrosis as a prime example of this.
Cystic fibrosis is fascinating from a biochemical perspective because it's often a case of the quality control system being too good, too strict. The most common mutation, the delta F508 mutation, causes the deletion of a single phenylalanine in a protein called CFTR, which is a chloride ion channel. This causes the protein to fold a little bit more slowly and less efficiently.
But it could still work.
Yes. Studies show that if you could get that mutated protein to the cell membrane, it would still function, maybe at 30% or 40% capacity, which might be enough.
Yeah, it never gets there.
It never gets there. The ER quality control system, the chaperones, and the ER machinery spots the slight misalignment during folding. It flags it as defective. It refuses to release the protein from the ER. Instead, it retrotranslocates it, tags it, and sends it to the proteasome for destruction. So the patient suffers from the symptoms of cystic fibrosis, not because they have a non-functional protein, but because the cell destroyed a semi-functional one before it ever had a chance.
That is tragic. It's bureaucratic perfectionism causing a devastating disease.
Exactly. And that's why a major area of therapeutic research is in developing pharmacological chaperones or correctors, drugs that can help the meat and protein fold just enough to pass the ER's inspection and escape to the cell membrane, where it can do some good.
On the other side of the spectrum, we have neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's.
This is the opposite problem. This is a failure of clearance. It's when the trash isn't taken out. In these diseases, specific proteins like amyloid beta and tau in Alzheimer's or alpha-synuclein in Parkinson's are prone to misfolding. They form sticky, insoluble aggregates. The proteasome can't digest these big clumps. They gum up the works. They accumulate inside or outside the neurons and are incredibly toxic, eventually leading to cell death.
And prion diseases are the extreme version of this.
The absolute extreme. That's where a misfolded protein acts like an infectious agent. A single misfolded prion protein can bind to a correctly folded one and induce it to misfold into the same toxic shape. It's a chain reaction, a domino effect of structural failure that spreads through the brain.
It's terrifying. And finally, Harper's mentions cancer. How does cancer exploit this system of protein governance?
Cancer cells are a mess. They're genetically unstable, rapidly dividing, and often in a hostile, low-oxygen environment. They produce a lot of mutated, misfolded, and unstable proteins. They are constantly under what we call proteotoxic stress. So to survive, they become addicted to the quality control machinery. They massively overproduce chaperones, especially one called HSP90.
Why HSP90?
HSP90 is a specialized chaperone that helps stabilize many of the key signaling proteins that drive cancer growth, the oncoproteins. Many of these mutated oncoproteins are inherently unstable, but HSP90 props them up and keeps them functional.
So they hijack the repair crew to keep their bad machines running.
That's a perfect way to put it. Cancer co-opts the cell's own survival systems, which is why HSP90 inhibitors are a major class of drugs being tested in cancer therapy. The logic is, if you take away the chaperone crutch, the cancer cell's unstable oncoproteins will collapse and the cell will die from overwhelming stress. You're attacking the cancer's support system.
It's incredible. We started this deep dive thinking about protein synthesis as just making things. But as we look back at the journey, from the strict logic of the triplet code to the high energy cost of fidelity, the solitary confinement of folding, the targeted shipping, and finally the regulated destruction, it really is a complete system of governance.
It is a dynamic, responsive, and incredibly expensive system. It invests heavily in verifying documents. That's the fidelity of translation. It provides escorts and security for VIPs, those are the chaperones and the SRP. And it shreds unauthorized or obsolete material without mercy. That's the ubiquitin proteasome system. This entire cycle ensures that the cell's protein workforce is potent, accountable, localized, and timely.
Governance in motion. I really like that theme. So as we wrap up, what's the one big idea you want the listener to walk away with?
I want them to fundamentally reconsider the energy cost of living. We often say we eat food for energy, and we picture running or jumping or thinking. But a massive, massive chunk of the energy you generate every single day from your food is burned just to maintain order, to proofread, to fold, to ship correctly, to destroy on time. Life is essentially an expensive, never-ending ATP-fueled battle against entropy. You are paying a constant, enormous metabolic premium just for organization.
And here's a final thought to chew on, something that wasn't explicitly in the sections but connects all the dots. Think about viruses. We've just spent an hour talking about how complex and expensive this machinery is. The ribosomes, the chaperones, the synthetases, the entire shipping and disposal network. Viruses, they don't have any of it. They are the ultimate biological hackers. They are just a piece of code, RNA or DNA, in a protein shell. They enter the cell with nothing but a frantic set of blueprints, and they hijack your entire bureaucracy.
They turn the factory against itself.
Exactly. They steal your trucks, your factories, your energy supply, and your shredders to build their own army. They exploit the very system of governance we just described. It's scary, but it's also a testament to how powerful and robust this system is. It's so good that even its enemies have to rely on it. All right, that's our deep dive into protein metabolism. I'm going to go eat some protein and appreciate the fact that my body knows exactly how to break it down and rebuild it into, well, me.
Just hope your chaperones and your E3 ligases are paying attention.
Fingers crossed. Thanks for listening, everyone. We'll see you on the next deep dive.